Good day everyone. Today I'm going to discuss to you the cytoskeleton and cell motility. But first, allow me to introduce myself. I am Joselle and Sumera and I'm going to discuss to you this topic outlines. So first, let us define cytoskeleton. So cytoskeleton is a dynamic and intricate network of filaments and tubules that spans throughout the cell, intricately weaving through the cytoplasm, the entirety of cellular material excluding the nucleus. So this structural framework provides essential support, shapes the cell, and facilitates various cellular processes, including cell division, intracellular transport, and maintenance of cellular organization. So the cytoskeleton's versatility and ability to undergo rapid assembly and disassembly contributes to its crucial role in ensuring the cell's structural integrity, mechanical stability, and adaptability to changing environmental condition. So ibig sabihin lang na ang cytoskeleton ay isang complex na network ng mga filaments at tubules na makikita sa loob ng cell at ito ay nagbibigay ng support sa shape ng cell at sa mga processes tulad ng cell division, transport sa intracellular at pag-maintain ng pagkakaayos ng cell. Next, we move on to the cytoskeleton filamentous structures. So, unang-una, we have the microtubules. These are long, hollow, unbranched tubes composed of subunits of protein tubulin. So, microtubule plays a crucial role in intracellular transport, cell division, and maintaining cell structure. Next naman, we have the actin filaments. The actin filaments are solid, thinner structure often recognized into a branching network. They contribute to cell shape, supporting microvilli on cell surfaces, and play a key role in cell movement and muscle contraction. Next, we have the intermediate filaments. So these are top rope-like fibers composed of a variety of related proteins. So they provide structural support and protection to cells, especially those subjected to mechanical stress. So, itong tatlo na to ay bumubuo sa cytoskeleton na siyang tumutulong sa pagbibigay ng shape, support at protection sa isang cell. Sunod naman natin ay ang properties of microtubules, intermediate filaments, at actin filaments. So, cytoskeletal filaments are dynamic structures that are constantly assembling and disassembling. This allows them to respond to changes in the cell's environment and perform a variety of cellular functions. So, microtubules are assembled from tubulin dimers, which are made up of two different tubulin proteins, the alpha tubulin and the beta tubulin. Microtubules have a defined polarity with one end, the positive end, growing faster than the other end or the negative end. This polarity allows microtubules to direct intracellular transport and cell division. Ngayon naman, we have the intermediate filaments that are assembled from tetramers, which are made up of 4 different protein subunits. So, intermediate filaments do not have a defined polarity and are not associated with the motor proteins. This makes them ideal for providing mechanical support to the cell. At susunod naman ay ang actin filaments are assembled from actin monomers. So actin filaments also have a defined polarity with one end that is barbed, growing faster than the other end that is pointed. So this polarity allows actin filaments to drive cell movement and muscle contraction. Ibig sabihin lang na iba't iba ang function ng mga bumubuo sa cytoskeleton. Una, ang microtubules na may dalawang tubulin protein, ang alpha at beta tubulin, at ito ay may defined polarity, ang positive at negative end, na mahalaga sa pag-transport at cell division. Sunod naman ang intermediate filaments na incorporated as tetramers, which ang tetramers is an oligomer formed from 4 monomers or subunits. Ito din ay walang defined na polarity at walang motor proteins na present sa kanya, hindi tulad ng microtubules na may motor protein at na kinesin at dyneins. So, ang huli naman ay ang actin filaments na may ATP actin monomer. Meron din itong defined polarity ng end ay barb na mas mabilis mag-grow at ang isa naman ay pointed. Next, we move on to structure and functions of cytoskeleton. So, microtubules transport vesicles and other organelles around the cell. For example, they can carry nutrients from the apical part of the cell to the basal part or transport waste or transport waste from the basal part of the apical part. So microtubules also contribute to maintaining the shape and structure of the cell. Intermediate filaments provide structural support for the cell, particularly in epithelial cell exposed to mechanical stress. For instance, epithelial cells in the skin are constantly exposed to friction and abrasion, and intermediate filaments help protect the cell against damage. Now, the actin filaments support microvilli on the cell surface. Microvilli has more projections that increase the cell surface area, allowing it to absorb more nutrients and secrete more waste products, and actin filaments also play a role in the cell movement. In neurons, microtubules transport organelles from the cell body to the axon terminals. This is crucial because neurons have long axons, and organelles need to be transported over considerable distances for proper functioning. So microtubules also contribute to maintaining the shape and structure of the neuron. Intermediate filaments in neurons provide structural support, especially considering the mechanical stress neurons often experience, such as stretching and compression in the spinal cord. So actin filaments are part of motile machinery involved in extending the neuron and cell division. Neuronal elongation is the process where neurons grow and develop their axons and dendrites, or dendrites. So cell division is a process where neurons divide to form new neurons. Actin filament plays a role in both of these processes. Now, during cell division, microtubules from the mitotic spindle aiding the segregation of chromosomes, microtubules attach to the chromosomes and pull them towards opposite poles of the cell, ensuring equal distribution in the two daughter cells. Intermediate filaments provide structural support during cell division, helping maintain the cell structure. Actin filaments contribute to the formation and contraction of the cleavage furrow, a construction in the middle of the cell that separates the two daughter cells. So, as we can see here, this is the cleavage furrow that's being seen. Next, we have our key takeaways. So, for our review, what are the functions of the cytoskeleton? So, the functions or the cytoskeleton serves as crucial role in the cell, providing structural support to maintain cell integrity and prevent deformation. It influences cell shape and motility through elements like actin filaments, enabling movement and migration. Acting as tracts, microtubules facilitate intracellular support or transport, ensuring the distribution of nutrients and organelles. In cell division, microtubules form the mitotic spindle, segregating chromosomes for accurate distribution. Additionally, the cytoskeleton aids in organizing cellular components and positioning organelles, contributing to the overall spatial arrangement and organization of the cell. So, now we are done to the overview of the major functions of the cytoskeleton. Next, we're going to move on to microtubules. So microtubules, composed of tubulin protein, are hollow and rigid tubular structures integral to various cellular components like the cytoskeleton, mitotic spindle, centrioles, and cilia, flagella, cores. So they play diverse roles, supporting cell structure and facilitating material movement between the cell body and neuron axon terminals. So microtubules consist of protofilaments formed by dimeric building blocks of alpha tubulin and beta tubulin subunits, just like what we have discussed a while ago. With a consistent polarity, the plus end is terminated by beta tubulin and the minus end by alpha tubulin. So non-covalent interactions between protofilaments maintain microtubule structure, and their structural polarity influence growth and directed mechanical activities. So, gaya nga ng sinabi ko, gaya kanina, na ang isang microtubule ay long, hollow, unbranched tubes composed of subunits of protein tubulin na mahalaga sa intracellular transport, cell division, and maintaining structure. Sunod naman, ito ay structure ng microtubules. So, the microtubule wall is sophisticated arrangement of globular proteins forming longitudinal protofilaments aligned parallel to the tubule's long axis. In cross-section, 13 protofilaments create a circular pattern within the microtubule wall. Protofilaments, the fundamental structure units, consist of dimeric building blocks formed by alpha tubulin and beta tubulin subunits, which tightly fit together in a three-dimensional structure. Non-covalent interactions between adjacent protofilaments are crucial for maintaining microtubule structure, contributing to its stability and integrity. Microtubules exhibit structural polarity, with all protofilaments sharing the same orientation, featuring an alpha tubulin at one end and a beta tubulin at the other. This results in a polarized polymer with plus N terminated by beta tubulin subunits and a minus N terminated by alpha tubulin subunits. So this is what it looks like in different looks. Now, we move on to protofilaments. So, protofilaments are crucial in the formation of microtubules, resulting to the orderly arrangement of tubulin dimers composed of the proteins alpha and beta tubulin. The polymerization of these dimers forms protofilaments, which align seamlessly to create a linear structure. These protofilaments interact to construct a hollow tube, a microtubule, which plays a vital role in cellular functions such as um, intercellular transport, maintenance of cell shape, and assistance in cell division. So this precise arrangement of tubulin dimers in protofilaments is a key step in dynamic assembly of an effective microtubule cytoskeleton component. So, dito naman sa protofilaments ay para sa pagbuo ng isang microtubules para mabigyan ito ng ayos at organisadong or organized arrangement ng mga tubulin dimers na may alpha at beta tubulin. So this is what a structure of microtubules looks like, which has tubulin dimer composed of alpha and beta tubulin. Pag sinabi kasi nating dimer, ito ay molecule or molecular complex consisting of two identical molecules linked together. So ang alpha at beta tubulin ay magkasama sa process ng polymerization at depolymerization, kung saan nagaganap ang microtubules nag-undergo extension and become faster or depolymerization, or undergoes shrinkage and becomes slower or depolymerization. Sunod naman ay ang MAPs or microtubules associated proteins, constitute a diverse group of protein within cell. So the term encompasses a heterogeneous collection of proteins with varying functions related to microtubules. The initial group identified, known as the classical MAPs, typically possess a structural configuration featuring one domain that binds to the side of a microtubule and another domain that extends outward like a tail from the microtubule's surface. This dual domain structure allows classical MAPs to interact with microtubules, influencing their stability, dynamics, and cellular functions. The classification of classical MAPs highlights their distinctive role in modulating microtubule behavior within the cellular environment. So, ang MAPs ay isang collection ng mga protein na may iba't ibang function, pero lahat sila ay connected sa microtubules. Ang isang grupo ay tinatawag na classical MAPs na may dalawang domain. Ang domain 1 na nagbabind sa isang side ng microtubule at ang isa naman ay umi-extend palabas tulad ng isang tail mula sa ibabaw ng microtubule. Sunod naman ay microtubules as structural support and organizers. So microtubules, due to their inherent stiffness, resist compression and bending forces. In a cell, the arrangement of cytoplasmic microtubules plays a crucial role in shaping the cell, much like an internal scaffold. So, gaya nga nung sinabi natin na ang microtubules ay nagtutulong o tumutulong sa pagbibigay ng shape sa isang cell. So, these microtubules function as structural supports and organizers, contributing to the cell's internal organization. Ibig sabihin lang, ang pinaka-function ng microtubules natin ay nagbibigay ng structural support sa isang cell para sa kanilang pagiging organized. This is evident in the fluorescent microscopy image of a cultured mouse cell where microtubules radiate from the peronoidal region following to the cell shapes, emphasizing their role in maintaining cellular structure. So, ang sinasabing yung tinutukoy natin kanina ay yung sa cultured mouse cell, ito yon. Yan. So, nagbibigay talaga sila ng shape para sa arrangement kung saan mapupunta yung kung saan malalagay yung isang cell. Okay? Sila ang nagsusupport ng arrangement at organization. Sunod naman, the microtubules plays a crucial role as agent of intracellular motility, facilitating the movement of vesicles between cellular compartments. This is particularly evident in axonal transport, the process of transporting neurotransmitter along the length of nerve cell axons. So the movement occurs bidirectionally, so away from the cell body and toward the cell body. Microtubules provide the tracks for this transport, and the vesicles carrying materials are attached to the microtubules by cross-linking proteins, including motor proteins like kinesins and dyneins. So this intricate systems enables the efficient and dynamic transport of essential substances within nerve cell, contributing a cellular function and communication. Meaning, ang microtubules ay mahalaga sa intercellular motility na patungkol ito sa paggalaw ng vesicles at ito ay may paggalaw na bidirectionally. Una ay ang away from the cell at isa ay towards the cell. So, para sa ating review, describe the three functions of microtubules. So, intracellular motility, unang-una, microtubule serves as tracks for intracellular motility, facilitating the movement of vesicles and organelles within the cell. So, yung microtubule, sila ang nagtatrack ng movement ng vesicles at organelles sa loob ng cell. They play a crucial role in transporting essential materials between different cellular compartments, contributing to cellular function and organization. So, sila yung nagta-transport ng mga kailangan nating materials sa different cellular compartments para magkaroon ng function at organization ng isang cell. Sunod naman ay axonal transport. In nerve cells, microtubules are integral to axonal transport, where vesicles containing neurotransmitter are transported bidirectionally along the length of the axon. So, gaya nga nung in-explain natin kanina na merong bidirectionally na mechanism. So, this process is vital for communication between the nerve cell, and it is facilitated by motor proteins such as kinesins and dyneins. So, yung axonal transport, vital siya para sa communication ng nerve cell natin para i-facilitate or fina-facilitate siya ng motor proteins gaya ng kinesins and dyneins. So, pangatlo naman ay structural support and organization. So, microtubules contribute to the mechanical stability and structural support of the cell. They resist forces that might compress or bend the cell. So, yung sa structural support and organization, ang microtubules nga ay nagbibigay ng stability at structural support para hindi siya magalaw, hindi siya ma-bend or ma-compress. Diba? So, similar to the way steel girders support a building. Kagaya ng mga steel na nagbibigay ng structural support sa mga building. At gaya din ng skeletal system na nagbibigay support sa body ng isang tao. So, the arrangement of microtubules within the cell helps determine its shape and internal organization, maintaining the overall structural integrity of the cell. Now, we're going to move on to the 9.3, the motor proteins, kinesins, and dyneins, gaya nga nung nababanggit natin kanina. So, kinesins, discovered in 1985, are motor proteins crucial for intracellular transport, initially isolated from squid giant axons. So, kinesin 1 is a tetramer with 2 identical heavy chains and 2 identical light chains. It features globular heads that bind microtubules, serving as ATP hydrolyzing engines, a neck, rod-like stalk, and a fan-shaped tail for cargo binding. Kinesins move along microtubules, termed plus end directed, and exhibit a hand-over-hand mechanisms. Kinesins are highly processive, adapt at long-distance transport, while most move toward the plus end, a subset, the kinesin 14, moves toward the minus end, another subset, the kinetin 13, induces microtubule depolymerization. So kinesins plays a vital role in organelle transport along microtubule tracks, influencing cell organization and function. So, ang kinesin ay ang nagmo-move sa microtubules na kung saan ang plus end directed siya sa mechanism na hand over hand. Samantalang karamihan ay nagmo-move patungo sa plus end, may isang bahagi or may isang part doon, yung kinesin 14, na umaandar patungo sa minus end. Ang isang bahagi naman yung kinesin 13, yun yung nag-i-induce ng microtubule depolymerization. Sunod naman natin ang cytoplasmic dyneins. So, discovered in 1963 as first microtubule-associated motor. It's a substantial protein responsible for cilia and flagella movement, comprising two identical heavy chains and various intermediate and light chains. It is a force-generating engine with a large globular head and an elongated projection or stalk. Okay? So, dyneins architecture and mode of operation differ from kinesin and myosin. It moves processively along a microtubule towards the minus end, opposite to most kinesins. So, cytoplasmic dynein plays essential role in mitosis, spindle positioning, and organelle transport, often working in concert with the adaptor dynactin. So organelles can bind both kinesins and dyneins simultaneously, allowing the bidirectional movement along microtubules. So the regulation of opposing motors and their potential tug-of-war dynamics are areas of ongoing investigation. Ibig sabihin lang, ang dyneins ay isang operation ay may ibang operation sa kinesin at myosin at ito ay gumagalaw sa microtubule papunta sa minus end, hindi tulad ng halos lahat ng kinesins. So, for our review, contrast the apparent roles of kinesin and cytoplasmic dynein in axon transport, and kinesin and cytoplasmic dynein, key motor proteins, and okay, so kinesin and cytoplasmic dynein is a key motor proteins in axonal transport. It exhibits distinct roles in cellular logistics. So, ano din siya? Yung process din siya ng pag-move ng mga motor proteins. So, kinesins, a plus-end directed motor, propels cargo towards the microtubule's positive end, facilitating the transport of organelles from the body to axon terminals. In contrast, cytoplasmic dyneins, a minus-end directed motor, moves cargo retrogradely from axon terminals towards the cell body. So, gaya nga nung sinabi dito, yung kinesin may plus end. Sa plus end siya pumupunta. Yung cargo pumupunta sa positive end na nagpa-facilitate ng transport ng organelles mula sa body papunta sa axon terminals. Yung cytoplasmic dynein naman, minus end directed motor. So, nagmo-move siya retrogradely or pabaliktad from axon terminals towards the cell body. So, kinesins typically transport membranous organelles such as mitochondria, while cytoplasmic dyneins is involved in retrograde transport of endosomes and lysosomes. Structurally, kinesins exist as a tetramer with globular heads acting as a force-generating engines, while the cytoplasmic dynein is a large protein composed of heavy, intermediate, and light chains. So, ang regulation nila ng mechanism ay iba. Yung kinesin, nag-exhibit ng plus and directed movement. Yung dynein naman ay nagpa-function in mitosis and spindle positioning. Kahit na opposing yung directions nila, yung kinesins and dyneins, nagko-collaborate sila, ensuring na may bidirectional transport doon sa axons na mahalaga para sa cellular organization and pag-function ng isang nerve cell. Yari na tayo sa motor proteins, kinesins, and dyneins. Ngayon, mag-proceed tayo sa microtubule organizing centers or MTOCs. So the function of microtubule in a living cell is intricately tied to its precise location and orientation. So understanding why a microtubule assemble in one specific place rather than another is crucial for deciphering its role within the cellular context. So, in vitro studies reveal that assembly of microtubules involves two distinct phases. A slow nucleation phase, where a small portion of the microtubule is initially formed, followed by a much faster elongation phase. However, within a cell, nucleation of microtubules occurs rapidly and is associated with special structures known as microtubules organizing centers. So among this, the centrosome is the most extensively studied in MTOC. Now, yun nga sabi natin na centrosome is yun yung pinagbabasihan o pinag-aaral mostly. Ngayon, i-discuss natin ang centrosome. So, centrosome is a siyang pivotal structure in an animal cell. And it is essential for microtubular cytoskeleton initiation and organization, consisting of two barrel-shaped centrioles surrounding by pericentriolar material or PCM. So, the centrosome recruits PCM to form a new centrosome where centrioles embed. With a nine-fold symmetry, centrioles nucleate microtubules, and during duplication, daughter centrioles form at right angles. Major sites of microtubule initiation, centrosomes polarize microtubules with -N associated with the centrosome, while some cells exhibit centrosome-centered microtubules, others like neurons or polarized epithelial cells show varied organizations. So centrosomes' absence in certain cells and genetic defects impact underlines their critical role, emphasizing the centrality of centrosomes in ensuring proper microtubule function and organization. So ano nga ba yung nabanggit natin na nine-fold symmetry? So, bawat centrioles ay naglalaman ng siyam na set ng triplet microtubules na nagbibigay daan sa characteristics ng nine-fold symmetry. Ang ganitong ayos ay mahalaga para sa pagbuo ng mitotic spindle na nagtataguyod or nag-help sa tamang pag-segregate ng chromosomes during cell division. Diba? Alam naman natin yon na yung mitotic spindle ay para magbigay ng equal na chromosomes during the cell division. So, ang centrosome ay nagiging sentro ng pag-organize ng microtubules na nagbibigay ng contribution sa structural stability at tamang pag-function ng cytoskeleton ng isang cell. So, very crucial ang centrosome kasi nga siya ang nag-initiate ng microtubules sa isang animal cells. So, responsible kasi siya sa initiation and organization ng microtubular cytoskeleton. So, ayan yung mga halimbawa. Ayan. Ito yung sinasabi nating pericentriolar material. Ito yun. Diba parang ano siya, diba double barrel. Ayan. Ayan. Sunod naman na ang centrosomes are not exclusively microtubule organizing centers in cells. So cilia and flagella's outer microtubules originate from structures called basal bodies situated at their base. So basal bodies, nag-share siya ng identity with centrioles at pwede silang magpalit or they are interchangeably transformed into centrioles. Halimbawa, ang basal body ng flagellum ng sperm ay nagmumula sa isang centriole ng mitotic spindle ng spermatocyte. At pagkatapos ng fertilization, karaniwang nagiging centriole ang sperm basal body sa unang mitotic division. Sa kabaligtaran, wala sa plant cell ang centrosomes, centrioles o malinaw na MTOCs. Sa halip, ang mga microtubules sa plant cell ay nag-uumpisa sa paligid ng surface ng nucleus at kumakalat sa buong cortex nito. Ibig sabihin, sa mga plant cells, walang malinaw na MTOC. Ibig sabihin, wala silang mga centers na nag-organize. Pero tulad nga doon sa may sperm cell na ang basal body ng flagellum ng sperm ay nagmumula sa centriole ng meiotic spindle ng spermatocyte. So, pagkayari nun ng fertilization, na nagiging centriole ang sperm basal body sa unang mitotic spindle. Sunod naman, ang microtubule organizing centers plays role in controlling microtubules dynamic in cells. So, kahit na meron silang iba't ibang mga itsura, lahat ng MTOCs nagre-regulate ng microtubule number, polarity, protofilament, composition, and assembly, timing, and location. So, ang karaniwan na sa mga MTOCs ay ang presensya ng white tubulin, isang critical na bahagi para sa microtubule nucleation. Ang white tubulin ay bumubuo ng maliliit na bahagi ng kabuuang protein ng cells, ngunit mahalaga sa pagsisimula ng microtubule assembly. So, sa loob ng pericentrolar material or PCM ng centrosomes, bumubuo rin ng ring-shaped complexes ang white tubulin or YTURCs na nagiging templates para sa microtubule nucleation. Ang YTURC ang nagdedetermine ng polarity ng microtubule at nagiging cap sa minus end na nagpipigil sa pag-aangat o pagkawala ng tubulin subunit. Ang mga mutations sa white tubulin ay nagre-resulta sa mababang microtubule nucleation na nagbibigay din sa mahalagang papel nito sa function sa centrosome. So, review tayo. We have what is centrosome, what component of centrosome nucleates microtubules, and how does it dictate the number of protofilaments. So the centrosome, a pivotal in animal cell, gaya ng sinabi sa atin, organizes the microtubule cytoskeleton with two centrioles surrounded by pericentriolar material. As a microtubule organizing center, the centrosome relies on white tubulin, a tubulin variant distinct from alpha tubulin and beta tubulin. So, ang white tubulin ay may crucial role that is manifested in the white tubulin ring complex or YTURC, ang isang helical array dictating the microtubule nucleation. So, ibig sabihin pala, ang white tubulin ay ang nagdedictate ng microtubule nucleation. The opening structure, the open ring structure formed by white tubulin and accessory proteins determines the protofilaments number in growing microtubules. So, the YTURC establishes microtubule polarity with the alpha tubulin at the minus end and caps the minus end, preventing tubulin subunits changes. So, protofilaments count is influenced by white tubulin abundance, emphasizing its central role in centrosomal microtubule nucleation and organization. So, para lang ma-clear, yung, di ba meron tayong alpha or yung A tubulin, the B or the beta tubulin, at yung Y, ito naman ay yung gamma tubulin. Next, we have the microtubule dynamics. So, microtubule in the cellular cytoskeleton ay dynamic polymers constantly undergoing assembly or polymerization and disassembly or depolymerization. So, yung microtubules, meron siyang pabago-bagong polymers. So, it is composed of tubulin protein subunits. Microtubules exhibit flexibility in lengthening and shortening process. So, itong microtubule daw ay may pagkakaiba-iba, nagbabago-bago siya. So, kasi pwede siyang mag-assembly at mag-disassembly. So, the stability of microtubules varies with mitotic spindle and cytoskeletal microtubules being labile or labile, neuron microtubules less labile, and centrioles, cilia, and flagella highly stable. And microtubules interacting proteins or MAPs and the positive TIPS are as well as the enzyme catenin influence microtubules stability. So, ang post-translational modifications tulad ng glutamate attachment sa tubulin ay yung nagre-regulate ng stability. Iba't ibang treatments gaya ng cold, pressure, at specific drugs ay nag-i-induce ng microtubule disassembly. At ang Taxol ay ang nag-inhibit ng disassembly at ginagamit sa cancer chemotherapy. Ang lability ng cytoskeletal microtubules ay nagmumula sa non-covalent protein associations na ito din ay nagbibigay daan sa depolymerization at repolymerization alinsunod or ang connected siya sa pangangailangan ng cells. Ang mga plant cell naman, nagpapakita sila ng dynamic microtubules array sa iba't ibang stages ng cell cycle. Nagpapakita ng rearrangement sa mga mechanism na disassembly and reassembly. So, the process of nucleation of plant cortical microtubules involves the initiation of new microtubule at specific sites on the surface of existing microtubules. In a live cultured tobacco cells expressing GFP-labeled tubulin, micrographs reveals an existing microtubule nucleating the assembly of a daughter microtubule resulting in a gamma-shaped, Y-shaped branch. The arrowhead indicates the end of the newly formed microtubule, while the arrow points to the branch point. So, electron micrographs of microtubules in a cell-free system shows two daughter cells or two daughter microtubules branching from the surface on an existing one. This process is schematically presented, highlighting the role of gamma tubulin present on the existing microtubules in the surface and nucleating new microtubules. Diba? Gaya nga nung sinabi doon na nagkakaroon ng dalawang parang nakasangay. Ayan o. Dalawang nakaganon sa isang microtubule. Okay? Yun yung two daughter microtubules na nagbabranch sa surface ng isa. Sunod naman ay ang the underlying basis of microtubule dynamics. So, the study of microtubule assembly and disassembly rates initially emerged from in vitro studies in 1972. So, Richard Weisenberg demonstrated that microtubules could be disassembled and reassembled by altering the incubation temperature. So, yung microtubules pala, pwede siyang ma-assemble at ma-reassemble dahil or sa pagbabago ng incubation temperature. So, sa vitro assembly, especially in the absence of gamma-tubulin ring complexes, may magre-result sa abnormal protofilaments number. So, yung mga maps ay at mga existing microtubules or structures containing them facilitates in assembly by serving as templates. So, ang GTP ay mahalaga para sa pag-assemble ng microtubule kung saan ang beta-tubulin ay kumukilos bilang GT phase. So, ano nga ba ang GT phase? Ang GT phases ay large family of hydrolase enzymes that bind to the nucleotide guanosine triphosphate or GTP and hydrolyze it to a guanosine diphosphate. Ang GTP hydrolysis na nangyayari sa pagkatapos ng dimer incorporation ay nakaka-affect sa structure at dynamics ng microtubule na nag-aambag sa inherent na instability na kilala bilang dynamic instability. Ang instability na to ay ipinaliliwanag ng dynamic instability theory na kasama ang pag-switch ng microtubules sa pagitan ng growth at shrinkage phases, lalo na ang kanilang plus ends. Mahalaga ang dynamics ng microtubules para sa cellular activities ang maaaring makita gamit ang technique tulad ng fluorescence speckle microscopy. So, the provided images depicts microtubules assembled in a laboratory setting. So, ayan. So in the first image, frozen and fixed microtubules that polymerize in vitro are observed using electron microscopy. So the individual protofilaments composed of globular tubulin subunits are visible. Notably, the middle microtubule in the image contains only 11 protofilaments, potentially due to the absence of proper template like the gamma tubulin ring complexes found in vivo. Now, the second image results or illustrates that in the vitro assembly of brain tubulin onto the plus ends of microtubules from a Chlamydomonas flagellum axonome, this process demonstrates how tubulin subunits are added to the existing microtubular structure, highlighting the dynamic nature of microtubule assembly and providing insights into their structural organization at the molecular level. Next, the structural cap of dynamic instability suggests that the growth or shrinkage of microtubule depends on the state of tubulin dimers at its plus end. In a growing microtubule, an open sheet with tubulin GTP subunits is present. As the microtubule grows, the sheet closes, leading to GTP hydrolysis. Once closed, only tubulin GDP subunits remain, causing outward curling and catastrophic shrinkage. So cryo-EM images illustrate this process. Microtubules dynamics in the living cells were observed in a fibroblast injected with biotinylated tubulin, revealing rapid assembly and disassembly within 1 minute. So next, this is the dynamic instability. And nagde-describe lang siya ng unpredictable switching between growth and shrinkage phases sa isang individual na microtubule. Ito naman ay binding of a microtubule plus end tracking protein. Ayan, plus end tracking protein. So, review tayo. What is the role of GTP in the assembly of microtubules? What is meant by the term dynamic instability, and what role does it play in cellular activity? So, ang GTP is crucial in microtubule assembly. Nagbabind siya sa beta-tubulin during the process. After incorporation, yung GTP hydrolysis ng beta-tubulin, nag-yield siya sa GDP, remaining in the microtubule and ensuring its dynamic nature. Ang dynamic instability naman ay proposed by Mitichon and Krishner, describes stochastic shifts between growth and shrinkage in individual microtubules. So, ibig sabihin, ang instability ay nagkakaroon lang or dinidescribe niya lang yung pag-shift ng growth and shrinkage sa isang microtubule, especially sa isang dynamic plus end. Ang phenomenon na ito ay mahalaga for cellular functions enabling rapid cytoskeletal remodeling. So, microtubules can swiftly disassemble and reassemble, gaya nga sa palagi nating sinasabi, na nag-i-influence siya sa process ng cell division, intracellular transport, at alteration ng cell shape. So, ang ability na nagtatoggle between growth and shrinking allows the cell to dynamically adapt their cytoskeletal structures to changing needs. So, pabago-bago para lang ma-cater niya yung changing needs. So, next, the structure and function of cilia and flagella. So, cilia and flagella are hair-like organelles found in eukaryotic cells, serving various functions like flow sensation, propulsion, and sensory roles. While referring to some same structure, yung term context-dependent akin to evening star and morning star for Venus. So, cilia likened to oars, more perpendicularly propelling fluid and particulate matter in coordinates bits. So, sa mga tao, yung cilia, yung sa respiratory tract, nakiklear siya ng mucus mula sa lungs. Ang mga non-motile primary cilia ay may mga sensory functions na nagmo-monitor sa mga katangiang extra cellular fluids. Yung flagella naman ay kadalasang iisa lang siya na nagpapakita ng iba't ibang patterns ng paggalaw, yung pag-move. At mga defect na cilia ay possible na magbigay siya ng iba't ibang sakit sa tao dahil sa pangkaraniwang papel na ginagawa ng isang cilia na merong sa motility niya sa paggalaw. So, cilia and flagella, commonly seen in cell biology, have a characteristic structure revealed in electron micrographs. Yung annexome, yung core nila, nagko-consist ng central pair forming the 9 plus 2 array. So, lahat ng axonemal microtubules, nagsasame sila ng polarity. So, the axonome includes components like radial spokes, interdublate bridges, and axonomal dyneins arms, responsible for generating bending motion. So, ang cilia or flagella nag-emerge sa basal bodies na kamukha sa centrioles. Ang basal bodies nucleate primary cilia ay non-motile or secondary cilia in a large numbers. So, ang growth ng axonome occurs at its plus ends, creating a construction site distance from the cell body. So, intracellular transport or IFT ay isang mahalagang process discovered in 1993 na nag-involve ng movement ng IFT particles na comprised of 20 proteins doon sa loob ng cells between the peripheral dublates and the plasma membrane na nagpa-facilitate ng cilia and flagella assembly. So, yung linear arrays na yun ay tinatawag na IFT trains transport na na yung particles niya um, to the cilium tip using kinesin, nagda-transport yung particles niya sa cilium tip using kinesin motors, while cytoplasmic dyneins brings them back to the cell body. So, yung mechanism na to na transport cargo protein such as the tubulin essential for assembly, inhibiting IFT, hinders cilia and flagella formation, aiding the study of their function. So, dynein arm acts na swinging cross bridges, generating forces for the sliding of microtubular dublates, leading to axonome bending and coordinated ciliary or flagellar movement. So, next-in linkages limit dublate sliding, and the central pair and radial spokes contribute to dynein and coordination. Although, yung central pair na crucial for signaling, yung motility ay possible in mutant flagella na wala ito. So, posing an answered question about the precise coordination of dyneins arms during the ciliary beating cycle. So, ito ay image na eukaryotic flagella in the unicellular alga chlamydomonas, Ray and Hardy. So, yung una ay algas 2 flagella visible, stained in a fluorescent antibody targeting a major flagellar membrane protein. Sunod naman ang structure of a ciliary or flagellar axonome. Ito naman ang longitudinal view of axoneme. Ang basal bodies and axonemes. At ang intraflagellar transport or IFT na merong two rows of protein particles na nag-i-indicate ng arrowheads, position between the outer dublae microtubules and the flagelloplasma membrane. Ito naman ang schematic representation of the forces that drive ciliary or flagellar motility. So, yung mga dynein anchored along the A tubule or alpha tubule of the lower dublae attach the binding sites of the beta tubule to the upper dublae. So, review tayo. So, contrast the sensory and motile functions of cilia and flagella. Describe how failures of these different ciliary functions contribute to human diseases. So, una, sensory functions. Cilia. Many cells in the human body have non-motile primary cilia na nagpo-function as antennas. So, nagko-contain sila ng receptor at channel na nagme-maintain ng mechanical and chemical properties ng extracellular fluids. Yung mga specialized sensory cilia, nag-i-include sila ng sensing fluid flow sa kidney, sa olfactory cilia sa nose, and outer segments of rod and cone cells in the retina. Sa flagella naman, generally function ng cell motility. Sa mga ibang case, sa sperm cell, yung flagellum, nagpa-propel sila forward for fertilization. Ang mga functions naman nila, motile functions nila, ang cilia, large tufts of motile cilia move fluid and particulate matter through various tracts in multicellular. So, example, ciliated epithelium in the respiratory tract propels mucus and debris away from the lungs, yung pag-alis ng mucus. Sa flagella naman, involved in the movement of the entire cells. The beating motion of flagella sperms, such as in the sperm cells, propels the cells through its environment. So, nakakontribute din siya sa human diseases. So, sa sensory cilia dysfunction, ang mga renal disorders, sa retinal disorders, and sa mga motile cilia dysfunction naman yung respiratory issues, yung infertility. So, muna tayo sa huli, sa intermediate filaments. So, intermediate filaments or IFs are solid and branched filaments identified in animals cells with a diameter of 10 to 12 nanometer. They provide mechanical strength to cells experiencing physical stress such as neurons, muscle cells, and epithelial cells. So, hindi siya gaya ng actin filaments at microtubules. Yung IFs or intermediate filaments ay diverse siya, chemically diverse group, encoded by around 70 genes, divided into 5 major classes. So, sa maraming cells, ang IFs ay konektado sa iba pang cytoskeletal filaments gamit ang plectin, isang mahalagang dimeric protein na may mahalagang isoforms. Kahit na magkakaibang amino acid sequence, ang IF polypeptides ay may parehong structural organization. May gitnang rod-shaped alpha helical domain na inuikutan ng variable size globular domains. Ang alpha helical rods na may dalawang polypeptides ay magkakaroon ng spontaneous na interact upang bumuo ng rope-like dimer na may sukat ng 45 nanometer ang haba na may defined na polarity. So, intermediate filaments represent a structure diverse, categorized into 5 classes. The class 1 to 4 contribute to the cytoplasmic filaments, while the type 5 or lamins is integral to the nuclear envelope. So, present in animal cells, the IFs displays a radial distribution and are interconnected by the plectin, a fibrous protection acting as a cross bridge for IFs, microfilaments, and microtubules. So, yung pinaka-common na structure organization ng IF subunits ay nag-involve ng alpha helical domain flanked by variable globular domains. Sa pag-interaction nila, yung IF subunits na yon nagpo-form ng rope-like dimers approximately 45 nanometer long. So, ang intermediate filaments, nagko-constitute siya ng diverse group of structural proteins na vital sa pag-maintain ng cellular integrity. Ayan, yan yun. So, intermediate filament assembly and disassembly. So, IF assembly, tetramer formation from 2 dimers in antiparallel alignment, 8 tetramers lateral association forming a unit-length filament. Unit-length filament associated end-to-end to create elongated IFs. Assembly steps do not involve ATP or GTP and lack polarity. So, ibig sabihin yung pag-assembly pala ng IF ay end-to-end na nagka-create ng mas mahabang intermediate filaments at wala siyang ATP at GTP at wala din siyang polarity. So, ang characteristic naman niya, less sensitive to chemical agents, challenging to solubilize. Ang dynamic behavior naman ng IFs, injected keratin subunits incorporate rapidly into existing IFs. Incorporation occurs within the filament's interior, not at ends. So, ibig sabihin, yung nag-occur yung yung behavior niya doon sa loob ng filament's interior, hindi sa dulo. So, yung assembly and disassembly, nakokontrol siya ng phosphorylation and dephosphorylation. And next, ito ay organization of intermediate filaments within an epithelial cells ay nagde-depict ng schematic drawing. Ito nga yun. So, yung nucleus ng connection na to sa nucleus are facilitated by the proteins spanning both membranes of the nuclear envelope. Moreover, yung specialized sites of adhesion such as desmosomes and hemidesmosomes anchors IFs to the plasma membrane. Notably, IFs exhibit interconnections with other types of cytoskeletal fibers. So, gaya ng microtubules, MTS, microtubules, and actin filaments. So, ang connection sa microtubule at actin filaments ay pangunahing ino-orchestrate siya ng mga miyembro ng plaquin family ng mga proteins. Ano nga ba ang plaquin family? So, ito ay protein that associates with junctional complexes and the cytoskeleton. So, nag-include siya ng desmoplankins, enveloplankin, periplakin, and plectin, at madami pang iba. So, ang isang particular na halimbawa ay ang dimeric plectin molecule. Ito ay complex network na may connection na nagbibigay ng integration ng intermediate filaments sa kabuoang cytoskeletal architecture ng epithelial cell. At yung strategic na positioning at yung pagpe-play niya ng IF sa nucleus at plasma membrane, kasama yung interaction sa iba pang mga bahagi ng cytoskeleton, ay nagbibigay ambag sa structural integrity at cell at sumusuporta sa kabuuang functionality nito sa tissue. So, types and functions of intermediate filaments. So, yun nga, gaya sabi natin, yung keratin filaments in epithelial cells and neurofilaments in neuronal cytoplasm, nagpe-play sila ng crucial role in providing structural support, maintaining cellular architecture, and absorbing mechanical stress. So, meron silang mga diverse roles at functions na specifically, yung naglilink sila sa psychological and pathological conditions. So, yung neurofilaments, isa siyang type ng IF na implicated in neurodegenerative disorders like ALS and Parkinson's disease. So, yung mga studies na to nag-emphasize ng crucial role ng IFs in maintaining the cell's function. So, for our review, give some examples that reinforce the suggestion that intermediate filaments are important primarily in tissue-specific functions rather than in basic activities that are common to all cells. Pangalawa, compare and contrast microtubules assembly and intermediate filaments assembly. So, una, examples supporting the notion that intermediate filaments are primarily important in tissue-specific functions include the K14 gene deletion in mice, mimicking epidermolysis bullosa, simplex, and affecting skin integrity. So the essential role of desmin, a type of IF, in maintaining muscle cell structure and occurrence of related myopathies in human further highlights the tissue-specific function. Additionally, yung vimentin gene deletion sa isang mice, nagre-result siya ng minor abnormalities na nag-a-affect sa cells na nag-underscore at nagva-vary sa importance ng IFs across different cell types rather than in basic universally common activities. So, yung microtubule assembly and intermediate filament assembly, nag-iiba siya in several aspects. Yung microtubules kasi, dynamic structure siya nagpo-form by a polymerization of tubulin subunits na nag-exhibit ng highly organized structures with clear polarity na nag-undergo sila ng rapid assembly and disassembly na crucial for cellular processes gaya ng mitosis and intracellular transport. Pero yung intermediate filaments lack of defined polarity na nag-assemble sa diverse protein subunits na nagpo-form ng stable and less dynamic structures. So yung intermediate filament assembly, ito ay mas mabagal at continuous process na nagpo-provide ng mechanical stability to cells. Yung microtubules kasi, may specific role siya in dynamic cellular activities. Gaya ng cellular segregation, intermediate filaments contributes. Pero yung intermediate filaments, nagko-contribute siya sa overall structural integrity of the cells. Lalo na sa mga tissues na subjected sa mechanical stress. So, yung distinction na sa assembly and properties, nagre-reflect siya sa functional roles of microtubules and intermediate filaments within the cellular cytoskeleton. And we are done discussing all of our covered units. And I hope you learned something, and that will be all. Thank you.